If you want the definitive shortcut to understanding the biggest fish story on the planet, and I mean biggest literally, well, you've come to the right place. Welcome to the deep dive. Today we are cutting straight into a crucial piece of research from 2023. It's called Assessing Mesopelagic Fish Diversity and Diel Vertical Migration with Environmental DNA, and it's led by Annette F. Govindarajan and her team. Our mission today is basically to unpack why these fish living way down in the ocean's twilight zone, that's the mesopelagic zone, why they're so vital. And, maybe more importantly, how scientists are finally starting to figure out what they're doing down there using tech that honestly feels a bit like science fiction.
Yeah, it really starts with the sheer scale. I mean, these mesopelagic fish, they make up the majority of the world's fish biomass. The estimates are just huge, sometimes getting up towards 16 billion tons.
16 billion, that's hard to grasp.
It really is. And to put that in perspective, just one type, the bristlemouth, fit tiny little things in the Gynostomatidae family.
Yeah.
They're thought to be the most abundant vertebrates on the entire planet.
The most abundant vertebrate. Wow. Okay, so they're not just numerous, they're actually essential, like cogs in the planetary machinery.
Exactly. Yeah, their importance goes way beyond just the numbers. They are probably the main drivers of what scientists call the biological carbon pump. And the sheer scale of their daily lives, it directly impacts global climate modeling.
How so?
Every single day, they're actively speeding up the downward movement of carbon. They basically eat near the surface at night and then take that carbon down deep with them. So they move organic matter from those productive surface waters down into the deep, dark sea. If we want accurate carbon budgets, effective climate models, we have to know where these fish are.
And how many there are.
And how many, and crucially, how they move. That migration piece is key.
Okay, let's paint a picture of the environment first, the mesopelagic zone. We're talking, what, 200 meters down to 1,000 meters?
Roughly, yes.
Yeah.
It's incredibly vast, it's freezing cold, high pressure, and yeah, almost entirely unexplored.
And then there's this daily routine. They have diel vertical migration, or DVM. You mentioned it briefly.
Yeah.
So every night they swim hundreds of meters up towards the surface to feed, right, under the cover of darkness.
Mm-hmm. Safety in darkness.
And then as soon as the sun starts coming up, they make this frantic dash back down deep.
To avoid being eaten.
Exactly. Back down to the safety of the depths, minimizing their exposure to predators near the surface.
And that daily commute is what moves the carbon.
That's the pump in action. But studying this migration with traditional methods, like nets, it's a real nightmare.
Why is that?
Well, towing nets, things like the MOCNS system, which samples specific depths, it's just slow. It's inefficient. And often it severely underestimates the number of fish, especially the small ones or the fast ones or the ones that are just good at avoiding nets.
Okay, so nets aren't giving the full picture. The researchers needed something better, like a more efficient way to get snapshots of who's down there.
Precisely. And that's where environmental DNA or eDNA metabarcoding enters the picture. Right. The sci-fi tech. You just scoop up some water.
Basically, yeah. You collect water, and then you analyze the genetic material that organisms leave behind. Could be shed skin cells, waste, mucus, you name it.
But there's a catch, right? It's not quite that simple.
Not quite. What's really fascinating, and kind of a technical hurdle, is that this whole molecular method relies entirely on having a really good genetic database before you start. If you find a DNA sequence floating in the water, but you don't know which fish it belongs to, well, that data point is pretty much useless.
It's like having a library without a card catalog, you said earlier.
Exactly that. And the library, or the reference database for these deep-sea fish, was, frankly, incomplete, especially for the specific genetic marker they wanted to use.
So what marker did they choose?
Well, the standard DNA marker for general species identification barcoding is often COI, and they use that too. But for this kind of high-throughput sequencing, looking specifically for fish DNA in a mixed environmental sample, they focused on a shorter fish-specific gene called 12S. They used a particular set of primers called myfish to target it. The big problem was many of these important mesopelagic species just didn't have 12S sequences logged in the public databases like GenBank.
Ah, okay, so the library was missing key entries for the 12S section.
Correct. So before they could even really test if eDNA could track that diel vertical migration, the DVM, a huge foundational goal of this study was essentially to build that specialized dictionary. They needed to create a comprehensive 12S reference library specifically for these deep-sea fish.
Right, makes sense. So that leads us directly into their methods. They were out in the North Atlantic slopewater doing this work, and they had, what, 3 main goals for the research crews?
Yeah, 3 key targets. First, build that crucial 12S reference library. Second, compare what they found using eDNA from water samples with what they caught using traditional net tows. See how the methods stacked up.
And third?
And third, use this new eDNA tool with its new library to see if they could actually detect and track that diel vertical migration pattern.
So how did they build that reference library, that dictionary? You mentioned needing physical specimens.
They did. They had to go back to basics. Really rigorous taxonomic work. They collected actual fish specimens. Then they identified them meticulously using morphology, looking closely at the physical anatomy.
Old school science.
Very much so. And then, crucially, they validated that visual ID using the standard COI barcode sequence, double-checking their work.
Got it.
Only once they were absolutely sure about the species ID did they generate the corresponding 12S barcode sequence for that same fish. They ended up doing this for 80 unique mesopelagic species, covering 30 different families. It was a huge effort to ensure that the genomic data they generated was absolutely trustworthy.
Okay, so library built. Then came the field comparison. eDNA versus nets. How did they do that?
For the traditional net sampling, they use that MOC nest system we mentioned. It's a set of nets that can open and close at different depths so you know where you caught things. Yeah. But they only towed it at night, presumably when the fish were shallower and maybe easier to catch.
Right, targeting the actual physical fish present at that time?
Correct. Then for the eDNA sampling, they used a standard oceanographic tool called the CTD Rosette. It's basically a frame with sensors and a ring of Niskin bottles.
Those gray plastic tubes you see going over the side of research ships?
Exactly. They use those bottles to collect water samples, about 5 liters each, at specific depths. They did 2 vertical profiles during the day and 2 at night. And they were careful to sample well away from the dawn and dusk migration times to get clearer snapshots of who was where during stable day or night conditions.
Okay, methodology covered. Let's get to the results. Did the hard work pay off? Starting with the reference library.
Oh, absolutely. It was a huge success. They generated those new COI and 12S sequences for all 80 species. And notably, for 18 of those species, their 12S sequences appeared to be the very first ones ever deposited in GenBank.
So genuinely new data.
Genuinely new data. And it gets even better, actually, because they sequenced both markers, COI and 12S, from the exact same physically identified fish.
Ah, the ones they identified morphologically first.
Yes, because they did that, they were actually able to find and flag potential errors in the existing public databases. They found cases where sequences already on GenBank, even ones that looked very similar to their data, seemed to be linked to the wrong species name based on their carefully validated specimens.
Wow, so they weren't just adding knowledge, they were actually correcting the record too.
Precisely. It highlights the importance of that rigorous groundwork.
Okay, so that's the library. Now the big comparison. eDNA versus the nets. Did they find the same fish? Did the methods agree?
Well, interestingly, not entirely. And that's actually a really crucial insight from the study. The species lists generated from the eDNA water samples and the MOC nest net samples were often quite different.
Different. So one method is better than the other.
Not better, just different. What it really confirms is that these methods aren't competitors, they're complementary. Each one has its own biases and strengths. Relying on just one tool, whether it's nets or eDNA, will almost certainly lead you to underestimate the true biodiversity down there in the twilight zone. You need both perspectives.
Interesting. So what were some of the standout findings specifically from the eDNA data? Anything unexpected pop up?
Oh, definitely. The eDNA dataset detected species that the MOCDS nets completely missed. This included DNA signals from large, fast-swimming pelagic fish like bullet tuna, and perhaps even more surprisingly, DNA from 4 different species of marine mammals.
Marine mammals, way down there.
Apparently so. They found signals for True's beaked whale, spinner dolphin, bottlenose dolphin, and Cuvier's beaked whale in various deep water samples.
That's incredible. Just from a 5-liter water sample, you get evidence of a beaked whale swimming nearby, potentially hundreds of meters down.
It's pretty amazing, yeah. But the real scientific jackpot, the main goal, was confirming they could detect diel vertical migration with eDNA.
And could they?
Absolutely. The eDNA data provided really powerful quantitative evidence for DVM. It worked. If you look at the shallowest depth range they sampled, 0 to 100 meters, the eDNA detected 13 different fish species in the nighttime samples.
Okay, 13 species near the surface at night. What about during the day?
During the day, in that same 0 to 100 meter zone, that number plummeted. They only detected 4 species.
Wow, 13 down to 4.
Exactly. That stark drop is the genetic signature of the migration. It's the fish making their daily dive down into the depths to escape the daylight.
And this result also validated their choice of the 12S marker, didn't it? You mentioned earlier studies using a different marker, 18S, on maybe even the same samples.
Yes, that's a critical point the authors make. Previous work using the more general 18S ribosomal RNA marker on, reportedly, the same water samples had failed to detect a clear DVM signal for fish.
So why was 12S the game changer here?
Well, the 12S gene, especially when you amplify it using those me-fish primers they used, is highly specific to vertebrates, particularly fish. The 18S marker, while useful for broader eukaryotic diversity, is just too general for this specific question. It often gets overwhelmed by background noise, huge amounts of DNA from tiny zooplankton, protists, other non-fish organisms.
So the fish signal gets drowned out.
Exactly. The 18S signal gets polluted, and you lose the subtle fish patterns. Using 12S gave them a much cleaner, laser-like focus, specifically on the fish DNA.
Okay, that makes a lot of sense. So thinking about the bigger picture now, the broader implications, especially those unexpected predator detections, the whales and tuna, what does that tell us?
It suggests a really exciting future for using eDNA in trophic ecology, studying who eats whom. Well, all 4 of those predators they detected, the whales and the tuna, are known to feed heavily on mesopelagic fish or the zooplankton found there. And the fact that the DNA signals from the tuna and most of the marine mammals were concentrated in samples from just one specific night sampling event, Cas9,
Yeah.
that strongly suggests that eDNA analysis might have the potential to pinpoint actual foraging hotspots in the ocean. You could potentially map where predators are actively feeding based on the DNA they leave behind.
That would be revolutionary, linking predators and prey just using water samples.
It could be, yeah. A new way to understand those dynamics.
But we should probably also talk about the limitations, the caveats. The study itself noted pretty high variability between duplicate eDNA samples, right? Even when taken at the same depth, same time.
Yes. They were upfront about that. High variability between replicate samples.
Which I guess makes intuitive sense if you're only working with a tiny 5-liter bottle of water in a vast ocean zone. But doesn't that kind of undermine the method? If 2 samples from the same spot give different results, how reliable is it for studying this huge mesopelagic zone that's, hundreds of meters deep and thousands of kilometers wide?
That's a really fair question, and it gets to the core challenge, the constraint of the method right now. The researchers acknowledge that variability, but I wouldn't say it undermines the method entirely. It defines its current limitations, especially for quantifying abundance precisely or getting a complete species list from one small sample. What the results do show is that even with that variability in specific species detection, the overall signal, like the massive DVM migration pattern, was still clearly and robustly detected. That big biological signal came through despite the noise.
Okay.
But yes, absolutely. To get a truly representative picture of all the species present and their relative numbers, 5 liters is minuscule compared to the sheer volume of the midwater environment. It's a sampling challenge.
And they also saw those other weird detections, right? Like finding bullet tuna DNA down at 775 meters. That seems way deeper than usual for a surface fish.
Right. And that points to the second major limitation, or maybe ambiguity, of eDNA. It tells you the DNA is present, but it can't always tell you the source form or context. Meaning, was that tuna DNA from a recently sloughed skin cell indicating the fish maybe was diving unusually deep to forage? Or was it from something else, like maybe an egg that sank or a fecal pellet sinking from predators that ate tuna higher up?
Yeah.
Or just dead cells drifting down?
Ah. So you don't know if it was a live fish at that depth or just its genetic echo?
Precisely. You can't always distinguish the source form, cells, gametes, waste, or know the life stage of the individual it came from, or even if the DNA particles simply sank from shallower water.
That ambiguity also ties back to the library issue, doesn't it? You mentioned earlier that even after building their library, not everything they found in the water samples could be identified. They used the term ASVs.
Yes, amplicon sequence variants, or ASVs. You can think of an ASV as basically every unique genetic sequence they detected after the sequencing process. Each distinct DNA string is an ASV. And the researchers found a large number of these unique ASVs that still remained unidentified. They didn't match anything in the public GenBank database, even after the team added their own 80 new validated sequences.
So even with their library improvements, there's still a lot of unknown DNA floating around down there.
A lot. It just underscores how vast our knowledge gap still is for deep-sea biodiversity. many species likely still lack reference sequences.
So looking ahead, the path forward seems pretty clear then. Address the sample volume issue and keep building that genetic library.
Exactly. To tackle the variability, future studies really need to incorporate much larger sample volumes. We're talking tens of liters, maybe even hundreds, per sample. This likely means using things like autonomous underwater vehicles, AUVs, or other uncrewed platforms that can carry large volume pumps and filter water in situ right there in the deep sea, concentrating the DNA from a much bigger parcel of water.
More water, less variability, hopefully.
That's the idea. And alongside that, continued dedicated effort to collect specimens, identify them carefully, and generate those high-quality reference sequences, both COI and 12S, is absolutely paramount. We need to be able to put names to all those unidentified ASVs.
And ultimately, eDNA isn't a magic bullet on its own, right? It needs to be combined with other methods.
Absolutely. The real power comes from integration, combining eDNA data with traditional net sampling, with acoustic surveys that can map fish layers, maybe even with video from cameras on ROVs or AUVs.
Yeah.
Putting all those pieces together, the DNA, the physical samples, the sound data, the images, that's how we'll eventually paint a truly complete picture of these critical mesopelagic ecosystems and food webs.
So summing up this deep dive then, this research convincingly showed that 12S eDNA metabarcoding is a genuinely powerful tool. It can observe mesopelagic biodiversity. It can track that massive diel vertical migration.
Yes, it's validated now. And it works best as a complement to traditional methods like netting, offering a rapid, non-invasive way to get a sense of what's arguably the world's largest fish biomass.
Which leads us nicely to a final thought for you, our listeners, to mull over. This study established that even a single tiny 5-liter water sample could reveal not just that huge daily migration pattern, but also fleeting evidence of large mobile predators like beaked whales, hundreds, maybe thousands of meters deep.
And the modeling work cited in the paper suggests that eDNA signals, once they're shed by an organism, tend to stay relatively localized, at least vertically, maybe within about 20 meters or so of their source before dispersing or degrading too much. So here's the question. Given that localized signal, and considering the absolutely enormous three-dimensional scale of the mesopelagic zone globally, how much sheer undocumented biodiversity are we still missing? Missing simply because the number of samples we currently take is so limited and the volume of each sample is so incredibly small compared to the environment itself. It really suggests the deepest secrets of the ocean are still down there, waiting for us to build a better dictionary and perhaps bigger buckets.